this is Farhad Tafezi from the ELSA Institute in Zurich, Switzerland. I am part of the team that implemented the original corneal cross-linking technology into ophthalmology back in the early 2000s. Besides my work as a cornea specialist and laser surgeon seeing keratoconus patients on a daily basis, I also run a basic research lab on ocular cell biology since 7 years. And one of the questions we have looked at lately was the effect of repeated cross-linking on ocular biomechanics. Now, this is an important clinical question. Why? Because everything we do in biology follows a so-called Gaussian distribution. So people do not always react the same. Most of the people would react the way we expect, for example, after corneal cross-linking with the stabilization of keratoconus. But then there is almost always extremes on either end. So people that react much more than we anticipate, which means normal flattening, but there are also patients that would react with a massive flattening. We have published on that back in 2010, people that flatten their cornea up to 10 or 11 diopters after a regular epithelium of Dresden protocol cross-linking. And then there are people that do not respond to cross-linking at all, so that show failures. So apparently the individual response of ocular biomechanics to corneal cross-linking can be very different. And if I had to cross-link again in the past 10, almost 15 years, these were often people with either a pellucid marginal degeneration, where the ectatic process happens in the lower periphery of the cornea, or if there was another additional factor that comes into play, like increased estrogen levels during pregnancy. Now, if we look at the cross-linking procedure, then there are 3 waves of reaction within the tissue. There is the photochemical reaction that happens very fast, within microseconds to milliseconds. And then within seconds to minutes, these covalent bonds between the proteoglycans of the cornea and the collagen side chains are formed, the crosslinks per se. And then there is a 3rd reaction that takes weeks to months, which is conformational changes of the collagen fibers and changes in the 3D structure of the cornea itself. And this leads to flattening. Now, what has been done so far in the laboratory? There was an IOVS paper from 2014, which was performed ex vivo, which means in porcine corneas. Once the animal was deceased, the cornea was taken, and then the cornea was subjected to more than one cross-linking. And these colleagues measured the biomechanical reaction of the tissue using a technique called scanning acoustic microscopy. And what they found was that if you were to perform 2 cross-linking procedures within 24 hours, then there is no measurable increase in biomechanics after the 2nd cross-linking. Of course, this is a simulation of what we do in clinics, because in clinics we would not cross-link within 24 hours. We would sometimes have years in between. Let's say a patient comes in for progressive keratoconus, receives a cross-linking, and after half a year we detect, progression has not stopped, and we would repeat the procedure. So there is not 24 hours in between the 2 procedures. There might be several months. And this is why we could only gain limited knowledge using this method. What we needed was an in vivo method, a method in the living animal. And this is why we have performed almost 2 years of work to bring the animal model of corneal cross-linking from the rabbit eye into the mouse eye. So we were able to cross-link mice and published this back in 2015. And what we have seen here in these mice, this is an anesthetized mouse that undergoes cross-linking with one of our MD PhD students. And what we have learned here is to detect the biomechanical changes in a very reproducible manner. And then we used this new knowledge to study the effect of repeated cross-linking once the cornea has reepithelialized with 1 week apart. And what we found here was quite surprising. If you see these curves indicate the stiffness of the cornea. The red line within the yellow curve is a control, which means this is not a crosslink cornea. This is a cornea that just only received riboflavin but no UV light. So this is our baseline. And you can see there is a certain biomechanical resistance of the tissue, a natural resistance. And then you have the red area with two lines in there. And these two lines are one crosslinking or repeated crosslinking performed. And these lines are so close together that there is no statistically significant difference between them. In other words, whether you crosslink once or crosslink, wait a week and crosslink a second time, there is no further increase in corneal biomechanics. What does this mean for the clinical condition? It means that if we were to redo a crosslinking soon after a first crosslinking, we will probably not achieve a further increase, so the effect is already maximal in the first session. But under clinical conditions where I see a patient after 2 or 3 years and then see a slow decompensation, a part of the original crosslinks have already been degraded. We know that there is a turnover in the collagen that roughly turns over the entire collagen content of the cornea within 6 to 7 years. So if I were to see a patient after 6 years with a progression of keratoconus, there are no previously induced crosslinks in this cornea present anymore, and the second crosslinking would, with great certainty, re-improve biomechanics. So again, this is very good to know, but we have to dig further into it, especially because we have some very limited, but we have some clinical data from humans that suggest otherwise. This is just a case report on a single case, but a very interesting case that I've published last year. It's a patient that I have cross-linked almost 10 years ago. As you know, we started in Zurich in 2004, and this was a patient we cross-linked in 2008 with the technology we built at the time. And as you can see, I followed this patient between 2008 and 2014, and on the right eye, the patient showed not only stabilization of the disease, but even an improvement and flattening of the cornea. Now, the left eye was also cross-linked in 2008 and behaved very differently. In the left eye, I saw a distinct change between 2008 and 2012 when the patient showed up for his 4-year follow-up control. And based on these changes, I decided to re-crosslink the patient after 4 years, performed a second cross-linking, and the patient showed a massive second effect, as you can see in the upper right. So in this particular patient, a second cross-linking was highly beneficial to ocular biomechanics or to ocular curvature. Taken together, there is a certain conflict between laboratory and clinical data. We will keep on looking further into this. The next clinical steps are to collect more data on repeated cross-linking, and in the laboratory we will perform cross-linking now with half a year apart to better simulate the clinical condition. This is Farhad Tafezi from the Elsa Institute in Zurich, Switzerland. Thank you for your attention.